Welcome. I'm Dr. Vinay Prasad. I'm a hematologist-oncologist, and I'm Associate Professor of Medicine at the University of California, San Francisco. In my professional life, I see patients, I teach trainees, and I do research in healthcare policy. This is Plenary Session. Plenary Session is a podcast at the intersection of medicine, oncology, and health policy, and you're listening to Season 3. On this week's episode. This week on Plenary Session, I'm joined by Arye Elfenbein. Arye Elfenbein is a physician, he's a cardiologist, and he is a doctorpreneur. He's launching a new company, and their goal is to make Ex-Vivo fish meat for human consumption. You won't want to miss this fascinating discussion about a San Francisco startup legend. If you like this podcast and want more content, follow me on Twitter at vprasadmdmph. Check out the YouTube channel, Vinay Prasad, MD, MPH. Patreon backers will get access to the slides for lectures I give on Plenary Session. Want to hear from us? Email us your question at plenarysessionpodcast@gmail.com. I'm back in Plenary Session HQ, joined by Arye Elfenbein. Dr. Elfenbein is a cardiologist, and he is, in fact, a biotech entrepreneur. Dr. Elfenbein, it's a pleasure to have you on the podcast.
Thank you for having me.
We were just talking about how good these microphones sound.
They sound amazing.
Amazing, yeah. These are the Shure microphones. Yeah. I'm still getting everything connected here. I don't know what all these knobs and buttons do. I'm working on it.
The closer I can sound to Ira Glass, the better.
Do your This American Life intro.
From Chicago Public Radio. See, can't do it. This American Life, I'm Ira Glass. He does it so that the entire introduction is like one word.
Yes. He does it well. Well, I'm glad we were able to sit down. I know our time is going to be cut short in a little bit because you're a busy man. But I think we'll put out what we get, and then we will go from there. So why don't we start by telling folks a little bit about your background? You know, you and I, of course, we have a mutual friend. That's how we know each other. That's how we've known each other. But you're a doctor. You're a physician scientist. You're card-carrying cardiologist. And you are somebody who is an entrepreneur. You started a startup company. So I wonder if you might start by the startup company. What is your company? What are you working on? What's your elevator pitch? And then let's get back into, how you came to this unusual or, these days less and less unusual career path.
Yeah, I mean, I'd start by saying that this is something I never thought I would be doing as a career. And so the company is called Wildtype. And the idea, just like the word Wildtype, is to create something as it exists in nature. And it really started from a trip I made back to Australia, where I grew up. And I was in the far north and looking around at a lot of the land that used to be rainforest and is now used for cattle farming, and thought about, what the kind of ecological impact is of our food system. And around the same time, I was inspired by the work mostly out of Shinya Yamanaka's lab. He
stem cell biologist.
Right. He was the one credited for discovering how to reprogram cells into iPS cells, pluripotent stem cells.
Pluripotence, yeah. And what was it? It was a couple of things he did, a couple of genes that he flipped.
Yeah, it was 4 factors, and they're called the Yamanaka factors. And that was at Kyoto University in around 2006. And I was there for grad school for my PhD around the same time. And so it was a very inspiring moment. And it made me wonder, with all of the advances in stem cell biology, do we need animals to produce meat? Could we just create the exact same thing just outside the animal from their cells? And I was, sort of driving through Australia and thinking these thoughts, and of course thought I was the first person to have this thought, and then went home and worked the Google machine and realized that actually many people have had this idea, and even Winston Churchill had
really
opined about this in the 1930s.
Really, I didn't know that.
About why one day we will escape the absurdity of growing an entire bird, an entire chicken, just to eat sort of part of it, and I was really fascinated by this idea and realized that another cardiologist, Dr. Mark Post, with whom I had actually overlapped in the same lab doing research in the early 2000s. He had spent the last few years working on this exact same thing and created the world's first hamburger in this way. I couldn't believe it. And the first conference, first international conference, was going to take place in the Netherlands soon after that. And I said, oh, I've never been to the Netherlands, and
you gotta be there.
Got to reconnect with Mark Post, and I don't know why I'm going, but I just went. I was riveted, and it really changed everything for me. I was a cardiology fellow at the time, and people, I said, I'm going to this conference, and people said, oh, like, is it the ACC, or is it? I'm like, no, people are making meat from cells. And
and they were like, okay.
Right. And it was such an interesting group of people. There were scientists, there were anthropologists, there were venture capitalists, there were people who were just enthusiasts, like futurists. I mean, it was a fascinating group of people, and I sort of kept this idea with me until I finished training, and then moved to San Francisco to work at the Gladstone Institutes at UCSF. I was working with Dr. Deepak Srivastava. And I was making these beating heart tissue little patches, and was thinking the same thing. Like, could
you were getting hungry?
Could we? Exactly. I'm like, this looks tasty.
Yeah.
No,
no, of course not. But you were thinking, can we find a way outside of an animal culture, outside of farm-raised salmon, to grow fish and fish meat?
Exactly. And so, seafood in particular, I think the kind of ecologic impact that both overfishing and even fish farming or aquaculture have had, those have been profoundly devastating in many ways. But not only that, I think that our seafood today, there's a big problem of traceability. A lot of what we buy, we don't actually know if it's the same thing.
I hear if you like DNA sequence it, it's like not even the fish you think it is.
Right. There have been quite a few exposés written about just that, about sort of ordering one thing off the menu in all kinds of restaurants in New York or LA, and finding out that that's actually not what it is. And part of that problem is an enormous part of the fish that we even catch here goes offshore, often to places like China, where it's cheaper to process the fish, and then they come back here.
I see. So your elevator pitch is it's about a 250 floor building. That's your elevator. Okay. But I want to say a couple things about you real quick. Okay, so one, you did your undergrad at Brandeis University, if I'm right. And then you did your MD PhD at Dartmouth University. After your MD PhD, you went to the fast track program at Yale. Where you did fast track in internal medicine. That's where we have our mutual friend. And then you went on and you stayed on for the cardiology fellowship. And then there have been some gaps in your training. There's a gap in your graduate school where you spent some time in Japan. I think listeners also have heard you allude to, but you spent some time growing up in Australia many years of your life. And I think the other very interesting thing about your journey is in your research portion of your cardiology fellowship. That's when you came to San Francisco and you started working at the Gladstone. And so you're a cardiologist. I think so. I should say all these things about you. You're a cardiologist, you're a physician scientist. Your company is seeking to make fish outside of actually growing fish and farming fish and catching fish, but having the fish meat product and have it taste as rich and you'll talk about that as the real thing. But you're also, every few nights a month, you're moonlighting as a cardiologist. You're moonlighting as a critical care doctor in the Kaiser network. So you're living the startup dream. Is that accurate to say?
I am, and it's a bit of a stereotype these days, I think, in San Francisco. But I will say that it's been an amazing journey. The sense of teamwork in a startup from the very beginning. When you start was just me and my co-founder, and then we had one other person, and then two other people, and it really just feels like being on this little life raft, and we get a little bit more secure on that life raft, and then we have like this little boat, but it's still very rocky, waters. And
I guess like one of the things that draw people to medicine is that it is I hate to say this, but to some degree it's a risk-averse career. People always say to me that, if I wanted to be rich, I would have been an investment banker. I wouldn't have been a doctor. And I was like, well, you're not thinking about that in the right way. The best investment banker clearly out-earns the best doctor by an order of magnitude. But the worst doctor probably out-earns the worst investment banker. Medicine is a profession where the person, in the 10th percentile, the 5th percentile, they're doing very well on average. It's sort of a safety net. And that encourages a certain type of phenotype, the type of person who always, I'm sure you're this type of person to some degree, gets good grades and is very sort of risk averse. Then entrepreneurial work is the opposite, right? I mean, you're playing a high stakes game that it's very possible, I hope it's not the case, but it's very possible that the company doesn't take off.
It's very likely.
Very likely.
Just by statistics. Yeah.
Yeah. So I guess, was it different for you, or were you always sort of somebody with an appetite for risk, as evidenced by the types of things you were studying as a researcher? How did it fit with your personality and your tolerance of risk?
Yeah, it was. I mean, I will just start by saying, even a career in let's say academic medicine is one that is very risky. You're right. I mean,
yeah, I know. Tell me about it.
Your trajectory is really defined by discoveries that you made. Like, these aren't milestones that you can just sort of plan out. And so,
well, with the right graduate student and the right Photoshop.
Fair enough.
No, but your point's well taken. Like, you can't, you can be a great scientist, but you don't stumble across something wonderful.
Right. Yeah. Exactly. I mean, if we were to ask Professor Yamanaka about what it took to discover iPS cells,
yes.
Obviously these are very risky things to be studying.
Yeah. So it's like timing, for serendipity,
right.
And if one were to we all know really good scientists who they don't have a Nobel Prize winning discovery in their CV, but they're probably just as smart, just as bright, just as talented as the person who did. They just happen to not be in the right field at the right moment at the right time.
Exactly. Or, their discoveries are just not perceived as quite as interesting at that moment in time.
That's where I'm at in my career.
So I think both are pretty risky, but I think for me, it just sort of reached a point of conviction where I really believed in the ability to create this, to be able to have cells grow in this way, and for a purpose that went beyond the biomedical sciences, that really influences our food system. And once I was at that point of conviction, it just felt, I don't know, it was kind of it was still a difficult decision to make to leave the university, but it was one that felt right. It's one that I knew that I would regret if I didn't.
Really?
And that, I think, was how I was able to make that decision in the end.
I see. So what you're saying is the thing that allowed you to tolerate the risk, to take the risk, was you felt like you had no real alternative.
I felt like, not that there was no alternative, but that I would probably regret the alternative.
I see.
And when I told Deepak about this, who's my boss, and he
started laughing. No, just
he didn't start laughing. I think it was certainly a surprise, but he said, the small decisions we make in life with our brains, and the big decisions we make with our hearts, and he said, I can see this is something that you care about, and this is something that you should absolutely do.
So you're telling me a story. I mean, you're motivated by passion. You're motivated by, I don't know what that motivation's like. I'm motivated by animus and spite and anger. Those are what motivate me. But you're motivated by passion. That's a much more commendable emotion to be motivated by. Okay, that's very interesting. So, okay, now this is what I really wanted to know. So I guess it's fair to say you're a doctorpreneur. That's what they call it.
I have not heard that.
It's a doctorpreneur. You're a doctorpreneur. In fact, I think it's gaining in popularity. But I guess one of the things I was curious about is, okay, so you have this idea. You have this kernel of this idea, which is we want to find a way to grow fish in sort of a cultured way that doesn't actually create the whole organism, but creates the product that we consume. I guess around this time, Beyond Burger was taking off Impossible Foods with Pat Brown and Mike Eisen and such. Did you obviously you're aware of their efforts, and how did you think about positioning yourself juxtaposed with all these sort of other big players, really?
Yeah, Justin is my co-founder, and Justin and I both sort of believed that there would always be a market for, not just a market, but there'd always be an appetite for something that is real meat in a way. We could have these plant-based alternatives, veggie burgers, as good as they are, and I love both Beyond Burger and Impossible Burger.
I see, but I see what you're saying. You don't consider it. It's not actually cells. It's It's a plant-based product with a whiff of hemoglobin moiety in it, right?
Exactly.
Some iron-containing moiety that gives you a little boost on the tongue, but it's not meat.
Yeah, and the difference when it comes to cells, this isn't just a sort of thing around semantics. It's that cells are programmed to create the same kinds of textures and structures, and encapsulate fat as adipocytes, or create the sort of parallel textures of myocytes. This is something that's very difficult to replicate when you work with plant-based products. And yet for us, growing cells, in a way, this is something that they are born to do. It's, literally in their DNA.
Literally, I see. So I guess while you were thinking about this endeavor, you thought that what's differentiating you from the competition really is that you're going to make more of a commitment to get this product to be closer to the thing that we've been, killing and trapping.
Right. We see this as really the next generation of all of these great plant-based alternatives right now, which is essentially, and I think there should always be the availability of fish that are fished responsibly by people who are stewards of our oceans and care about sustainability. I think aquaculture should always have a place in our food supply as well. But with the population just growing as quickly as it is, that demand is just far going to outstrip our ability to produce anything, right, to sustain it. And without a new source of meat or seafood, the kinds of scenarios that play out are either only the ultra wealthy have access to these types of foods, or we completely destroy the environment in the process of getting every last fish out of the ocean.
Right.
And neither of those sound great.
Yeah, and just to add to that, it's been a long time, but in college I took a class on like marine biology, marine ecosystems, and one of the things that it's been a while since I remember this, but is that that we talked about was that many people believe that farm-raised salmon, for instance, is not that bad for the environment. It's farm-raised after all. But then my understanding is that actually to farm-raise it, they go to a place in the environment that's very valuable and serves a key ecological role, and they destroy it to make said farm in said spot. And so it's actually not so good.
Right. This was something, to be honest, I didn't know. I didn't understand, until we started the company, is what aquaculture actually entails.
Yeah.
And, I think there are efforts now to make it better in some ways, but absolutely these sort of ecological dead zones, what they're called, are some of the most common issues, as well as issues of parasites and a lot of other. I think that the biggest problem though for aquaculture today is that the overwhelming majority of the food for these fish actually comes from other fish. So it's bycatch, from the ocean.
So they have to trawl the ocean to feed their farm.
Exactly. And so, if you believe that there's a problem with overfishing, then aquaculture actually doesn't solve that, because you are, as of today, the food that these fish require are actually just other fish.
I see. Oh, that's interesting. I didn't know that. Okay, so you have this seed of an idea, this germ of an idea, and you do have, to some degree, the technical know-how, how to start to think about how to do this. What's the next step in the life of a doctorpreneur? I mean, do you need the funding? Do you need? You must. Obviously, it's a resource-intensive thing.
Of course.
So what do you do when you have? Yeah, you guys have this idea.
Yeah, it started with a nights and weekends thing, and San Francisco was a unique place back then, 4 years ago, and we started in 2016. In that there were they're called incubators, which is kind of a funny word.
I've seen Silicon Valley.
Right. Where you can rent bench space, lab space by the hour.
Really?
And that's what we did, just kind of with our own non-savings. And it was just sort of this hobby and side project. But once we realized that there was only so far we were going to be able to get that way, we did go out and seek funding, and we sought funding from the NSF, which was not successful, and then also from venture capital, which was successful for us. There are certainly other sources of funding, and these were all ones that we had considered at the time. You can imagine for a technology like this that has a very long lead time.
Yes, very long, yeah.
And is very capital intensive upfront to just, what the R&D, a salmon cell line didn't exist in a 3D environment when we started.
Really? There was no salmon cell lines?
There were salmon cell lines from the 1960s and 1970s, actually.
Still salmon.
And they were grown in 20% fetal bovine serum, which is kind of a strange idea, when you think about it as a food, right?
Yeah,
you're using sort of the blood of unborn cows to grow fish cells.
Yeah.
And so, you can imagine what some of the, R&D milestones we had to achieve to create something more in the way that we think about for food.
So tell me about that. So you want to grow salmon cells. What are you growing them in? What broth?
Yeah, so the same nutrients you would find. Right. And, that involves obviously things like amino acids to make proteins, and then, carbon sources for energy like glucose or, other sugars, certainly fats, and then some growth factors that the salmon cells would produce ordinarily. But the expense of that is one that is, typically cost prohibitive. If we had made an entire kilo at the very beginning when we first started,
first generation technology,
it probably would have been about $500,000 a kilo.
Oh my God. That's a pricey night at the sushi restaurant. Yeah.
Right.
I've been to some expensive sushi restaurants, but not that quite.
Not like that.
Not like that. Yeah. I see.
And so, part of the reason was this was an inefficient system for growing the cells. Another one was we were using pharmaceutical grade reagents, not food grade reagents.
I see. That's a higher grade.
Exactly. Yeah. And there just wasn't a great system in place at the time to create food in that way. And that's still something that all of the companies in our space are really struggling with. Yeah.
Well, let me ask you this: when cells grow in culture, and it's been a long time, but I did do a few weeks of misguided work in cell culture. My understanding is that normal cells have contact inhibition, so when you culture them at a certain amount, they're just going to stop growing when they hit walls, when they hit barriers. Cancer cells, of course, notorious for sometimes they have inhibited contact inhibition. Do you have to inhibit contact inhibition in your cell lines?
Yeah, so there are a couple ways to do that. One is to select for cells that don't exhibit that, and in some ways have sort of changed their genetic expression profile to not require contact inhibition. But in the case of something like an iPS cell, going back to that, these cells are remarkable not just in the fact that they can become any cell in the body, but also because they double faster than pretty much any cell in the body. Their doubling time is so fast. And so these are the kinds of things that we look for when we sort of profile or genetically profile the different cell lines that we've developed.
It's very interesting. I want to come back to how you're doing what you're doing, but I want to just take a quick departure. I wonder about, I see these people, there are a lot of them, they don't seem to like science, apparently. There's a few more. You may know of such people.
I do.
I don't know. They say things like this mRNA vaccine and blah, blah, blah. It's going to change your genome. And obviously they don't know what mRNA is. Unless I've grown reverse transcriptase, I'm probably safe. They also say things like any GMO.
Right.
I hear that. I hear this. That you don't want GMO food. I was like, well, you don't want food that's able to grow? Okay, fine. What do you want to eat then? All right, well, fine. Okay. Or whatever. They don't want it you don't want to use genetic engineering to do it. You'd rather just breed it for a few 100 years to get what you want. Okay, whatever. I mean, they're this kind of mentality. Mm-hmm. So I guess my question to you is whether or not we are sympathetic to the view or not sympathetic to the view, we must acknowledge that these people exist. So what is the acceptability of the public? When you if you told the average Joe, I'm going to grow the salmon in this thing, are they gonna? I don't know what you must have studied some market study. What do they think?
Yeah, there actually have been a lot of studies about this, and first of all, it's obviously a strange idea for anyone. It's the first time they hear it because you're used to your meat or seafood you're used to the right. You're used to the idea that this came from a living animal.
Yeah.
That's the first thing. Often it does get combined with some sort of vague idea of genetic modification, which is a very fraught term. Obviously, I think that today it's come to mean something that is completely different from what gave it a bad reputation many years ago. In the days of Monsanto and so forth.
I see.
And obviously, creating this for the purpose of food has nothing to do with developing, resistant crops and, basically strong-arming monopolies and so forth.
Right.
And they're very different things. But still, in places like Europe, to introduce a product like this with any sort of directed genetic modification would just not be possible.
Really? They don't like
it's just right. Today in the U.S., genetic, like when things are declared as non-GMO, it's still kind of a self-declaration, but it's one that we recognize would just be another barrier, up front to consumer acceptance, and so it's one that our cell lines are currently non-GM. I mean, we've looked at GM and non-GM approaches. To your point, there's such similarity between those. I mean, we've been breeding everything that we consume over the course of centuries, and that on its own is some amount of, like, just look at the banana.
Yes, it's genetic engineering that's just done rather bluntly and not as thoughtfully.
Exactly. Yeah. I often will say, like, have you ever tried one of those seedless watermelons? Have you tried?
Yeah.
And people are like, yeah, of course. What do you mean?
Of course.
I was like,
a seed?
Well, how did that happen? Like, this seedless watermelon, clearly, like, that's not genetically normal, or else how would it reproduce, right? Like, it's things like this that we've come to accept. Yes. Why? There are all kinds of examples like this, but
I always find it. I mean, I guess I just get struck where, I don't want to take too. I guess I'd say no matter where you fall, one of the things I'm interested in is consistency. And when I see things every day in the store that people happily put in their mouths that don't even decay, I mean, things that you like left out on your counter, and 4 years later you will return and it's in the exact same form. The bugs and the bacteria won't even eat this shit, and you're gonna eat it, and then that they're gonna put in their mouth, but then they're gonna give you a hard time about, like, I don't know, some wheat that may have some thing in it that helps it avoid frost. I'm like, all right. Well.
And so, I mean, I think that to what you alluded to in terms of, like, for example, the new coronavirus vaccines, the mRNA ones, where people, understandably want to learn more about it, want to, see that it is safe. That's completely reasonable. But these are things that, first of all, me receiving my mRNA vaccine was actually a pretty emotional experience. mRNA in this way was exactly what I was working on at Gladstone and UCSF. In that case, it was injections in the heart, but it was really amazing for me.
But to be clear, that's not how you got your vaccine. Didn't get it all the way. Right.
I did not get it in the heart. I got it in my arm.
Okay, good.
But having made this with my own hands and like understanding everything that goes into this, like I obviously felt such a not just a level of confidence in its safety, but more was just amazed that it was so effective and was able to do so much good in such a short amount of time. And people have asked these very good questions, like, can it affect your own DNA? And that's a very simple one to address. Like, mRNA is not able to, doesn't even go into the nucleus. It doesn't interact with your DNA.
How dare you?
And this is something that sort of
and then let me ask one question. Like, the reason, my understanding is that, like, why did they even pursue mRNA? And the answer is you can do it very, very quickly. From the moment you know sequence, you can already you're already there. You can make your mRNA and you can go from there. You need to wait to get it to the cells and trans.
Exactly. The older way of making vaccines was to have a non-infectious component, the protein, the fully formed protein that often had to be folded in the right way, or a sort of weakened version of the virus, and you need to make sure to, like, weaken it enough that it can't kill it. But if you weaken it too much, then often it can completely degrade or be misfolded, and your body won't even recognize it as foreign.
And I've been reading about that, and my understanding is that it was, like, really a lot of trial and liberal artistry chemistry. It wasn't even science. It was like somebody just
Exactly.
eyeballing this embryo cells and like, that looks like the right amount of
Yeah.
cell death. That's the good shit. I got it right here. Yeah, it was really like just total, like
Yeah.
a baker in the kitchen.
Right.
Yeah.
But, pretty soon after the novel coronavirus, was recognized to be wreaking havoc internationally, we were able to get the sequence, I think it was as early as March. And we knew the spike protein was one of the most antigenic aspects of it. And from that point, it was very easy to just create that sequence and have the body recognize it as foreign and incite the body to then develop antibodies toward it.
And then the way the vaccine works, of course, is that the mRNA gets taken up by presumably your muscle cells, your skeletal muscle cells, but maybe whatever cells are in that vicinity.
Right.
Those cells express spike protein, they get killed off, and then the body learns to kill off spike protein.
Yeah, exactly. I mean, and that RNA just, over time just degrades and it's gone.
So you say.
So I've seen.
So you see, that's your story. Okay. All right. Very interesting. Okay, cool. So let's go back to your company. So your first few years, you're working at these rent a lab bench, like a
Right.
U-Haul. You're renting a lab bench only in this city, and you're trying to get this to go. Who are the first venture capitalists that you're deceiving? I mean, no, I mean persuading of your venture. Yeah.
Yeah. I think the theme was certainly investors who understood the long-term vision.
I see.
And, were comfortable enough with that amount of risk from such an early stage. And we're not looking to just quickly flip a company, because this is not the kind of company that that can really work that way.
I see.
And it took a lot of just sort of explaining what the vision is, a lot of what, my own fears were around whether this would scale, whether, costs would come, and just being very honest about those things, and then just describing what our vision was to address all of those. And it was really those kinds of conversations that led to investors who were really the most aligned with what we were doing. Who didn't just see the market size for all of food and, just kind of wanted to quickly cash out. But that's why, it's actually, I feel very lucky for the investors we've had along the way.
I see. And now 4 years later, what are your resources and where are you?
Yeah, we moved into our own space. We're a team of 20 people.
20 people? I didn't know that. That's cool.
Yeah. 2 of which are non-scientists. So we have a team of tissue engineers. We have a team of cell biologists. we have a team of media and process development scientists and food scientists.
Wow. And, just before we started talking, you were on a phone call. When this ends, you're going to have to go to a meeting. So it's a time-consuming thing. I mean, this is what you were trying to tell me.
Startup life is, it's pretty time-consuming.
It's time-consuming.
For sure.
So, I mean, tell us about that a little bit. I mean, obviously, there's the young medical student at home listening to this who says this is the way to get rich quick. You would say that's not the case.
Maybe a different kind of start. Maybe
Okay.
something like software. I think
Yes,
that,
okay.
Starting a cellular agriculture company is not a good way to get rich quick.
I see. Okay. Okay. But I want to get a sense of what is the work week like? What is your week like? And then tell us about we'll find the next thing I want to talk about is how you stay clinically relevant. But I guess what is walk me through a week in your life? Does it have normal business hours? Are you sort of always tied to this? Yeah.
Yeah. I think that the blur between work and life is more blurred in the early stages of a startup. But it's something that I've enjoyed, mostly because I love the people I work with. And there are so many different aspects to any day. I mean, one moment I'm on this wonderful podcast here.
The highlight of your week, obviously.
For sure, the highlight of my week.
Sure.
The next, we're preparing for board meetings or planning scientific experiments, or I still spend time in the lab and will passage cells, and for me it's an important thing to still be
What you doing that for?
Close to, I mean, because I think it's an important thing to be close to the data and to sort of like understand how these cells are growing and what it is that change. And it also gives me an excuse to try some of the crazier ideas.
You're like Henry Ford going to the assembly line one day and just building one himself, huh?
not quite Henry. Well, yeah.
It's kind of like that.
Yeah, no, I see. Yeah, I mean, but I also think it just sort of helps me understand where we are as a company as well, and so and not be too removed from what the experiments are. So it's all of those things. It's talking to investors. It's just sort of planning vision. There's some fundraising aspects to it. It's all of these things.
And there are things that would it be fair to say that there are a few things you're thinking about, like, I hope I get, and you could get a phone call on Monday and it could be the kind of news you're looking for. I hope I get somebody who works in your company could come tell you some news on Wednesday. There are a lot of those kind of hope for moments. Is that fair to say?
Yeah, absolutely. I mean, it's something that just every day is just a completely different thing. And, there's a lot of constantly putting out fires, which is exciting on its own. But just the fact that every single day is quite different. And if I'm doing the same thing, even a year from now than I'm doing now, then there's probably something that I'm doing wrong about it. It's not a very, it's not a job that should be static at this stage.
It's very, I see.
Yeah.
Interesting, because I mean, obviously, like in the career of an academic, you have these hope for moments, but I have noticed, and I've talked to some very senior people, that the further you get out, the less you have, because you're like, it could go this way, or if not, I don't really care. and that's a strange feeling. I mean, indifference is a strange feeling. When previously something would really be a great boost or a great setback. Now it's great with indifference. Okay, now let's talk about the clinical side. So you are a cardiologist.
Yeah.
So you know about aspirin, statin, beta blocker, ACE.
I do.
Those medicines, I feel like, I always joke with my cardiology friends, which is like, like oncology, like we have like a couple hundred drugs.
Right.
And you guys, aspirin, statin, beta blocker, ACE.
Yeah.
And then Entresto. You got your Entresto, and then you got your SGLT2 inhibitor, and then you got some things that inhibit platelets, and you guys run these like 800,000 person trial to find a slight difference in that in the platelet inhibitory strategy. Right. Okay. Anyway, I just tease. But you are a card-carrying cardiologist, and during all this, you have not given that up. I mean, you're still a cardiologist. You're still in KP in Kaiser Permanente, San Francisco, a few days a month. I wonder if you might talk about what that's like to keep a foot in the clinical arena.
Yeah, I mean, it's always felt like a privilege to be part of patients' lives in that way, and it's something that I haven't wanted to let go of for that reason. I think in many ways I probably enjoy medicine more now because it's something that I don't get to do as much. And so, I'll put on my scrubs and go to the ICU, and it's like, okay, we're here tonight. We're making this happen. This is great. And I love feeling that energy that maybe I wouldn't if it were 7 days a week at this point. But I think it certainly is more of a challenge to stay clinically relevant, as you're alluding to. This is a whole different part of my brain to kind of and I think the thing that drives that and the ability that I am able to sort of keep up to to the extent that I can is really just the curiosity of like just still being curious about what does this trial say, like what did
right
what's going on in the world of cardiology these days, and it's probably because it feels like a special thing to still be part of that world more than anything else.
That's very interesting. And I can totally see that. And I can also see, I wonder if you think about this, that these two things you do, they have very different horizons on which the outcome is related to the action. Mm-hmm. Critical care medicine is perhaps the shortest distance between you doing something and you seeing at least the surrogate of that action. Mm-hmm. Turn up pressor, turn down pressor, fix aortic balloon pump from inflating in the wrong moment. You know what I'm about to say.
Yeah, I know what you're going to say.
Yeah, so here's what I'm going to say.
I'm picking up what you're putting down.
Yeah. So of course, I think people tell me if I'm wrong. It's been a long time since I did this, but the principle of the intra-aortic balloon pump is that when the heart is contracting during systole, it deflates, creating a negative pressure in the aorta that allows blood to be pulled out of the left ventricle. When the heart is in diastole and the coronary arteries are being perfused, it inflates, which actually has a retrograde flow into the coronary arteries and gives the coronary arteries blood.
Right.
However, in actual practice, every so often, somebody has not been very attentive to the intra-aortic balloon pump, and it is actually inflating in systole and contracting in diastole. Is that possible?
It's possible. Let's just leave it at that.
Let's just leave it at that. Yeah, and I've seen that, and so when I finally saw the results of the randomized controlled trials of IABP in I guess in high risk 3-vessel stenting, complex stenting, and in shock, and some of those results are not as positive as people thought. I thought that one of the possibilities is it wasn't gated right, but anyway, that's a joke. So okay, but my point is, okay, so in the ICU, you're doing things and you're seeing the result of that every hour by hour, minute by minute, and then on the other side of your life, you're doing things and you may not potentially see the result of that for 20, 30 years. Let's not say 20 years, but maybe 5 years, 10 years.
Yeah. I think it's very similar to what medical school and grad school, the difference there, where I didn't know when I was going to graduate from a PhD. It just, felt like it was just going to
interminable
be the rest of my, like I was just going to die a grad student, like probably.
Oh, yeah.
As opposed to medical school where, you show up and you study and, this rotation ends on this date and so forth. And you know what the timeline should look like. And I don't know. I guess I've been comfortable with both of those.
You've
sort of
long had
things.
those two kind of risk-reward balances in your life.
Yeah, I think they do balance each other out. There is something more the sort of instant, like understanding, like seeing the results of your actions, and then the opposite extreme of like hopefully seeing that in 10 or 20 years.
Yeah. I guess, it's interesting when you say that because I wonder, in my case personally, if some of that long horizon is just something that I don't like, because both, obviously I did not do a PhD, particularly in the laboratory sciences, and if you made me do one, I don't know if I'd survive. And I obviously do a fair amount of clinical medicine, and probably even more in recent when I had my old job. And then the research I do is always very, compared to lab work, it's short time horizons. Like you have an idea and you'll get data back in a month, and you'll have the paper done within 5 months or something like that. And there's something to that. There's like, you like that kind of time span. The longest time horizon projects we did were some of these reversal projects where it took a year or 2 years. And that felt like an eternity. But in the lab, people do 10 years, easy.
Yeah, I think it becomes most difficult when, people are working for 4, 5, 6 years for one paper, and at the end it's sort of it's one, like everything, it's all the eggs in one basket. And
then it goes into PNAS and
what is wrong with PNAS?
Oh, come on.
No, exactly.
No. Yeah.
But I think it's an interesting point. And maybe we should try being a little more comfortable with those
long-term risk horizons. Oh, I don't know about me. Okay, you got to go in a minute. Last question before we'll pick this up some other day. And actually, maybe the thing I actually wanted to talk to you about, we didn't get to talk about. But well, that's for future episodes. This will introduce my audience to you.
Thank you.
And I'm sure they're going to like it because this show, what people say, the number one thing the show is missing is humor, and that's why you're here to supply.
Oh yeah, I have jokes.
Yeah, you got jokes. Yeah, we'll just stand up next time.
Yeah.
Okay, so the last thing I'll ask you before we'll pick up another day is, I don't know. I guess, what is your advice to the young person out there? I don't know. You must get young people coming to you and asking you advice, and I guess I can imagine. Maybe I'll just pick somebody. This is somebody who went to college like you, smart kid. I heard rumors. I heard you went to college pretty young. But smart kid, young person. They don't know exactly what they want to do in life. They feel like they want to make a difference. They want to be on this sort of intersection of, I don't know, science and medicine. Sort of a lot of the feelings that I think you and I must have had. They're debating physician scientist path. They're debating traditional MD path. They're debating just kind of laboratory work. Maybe they're debating just going to work for a company. They're debating being an entrepreneur, being a clinician, all the sorts of things that you kind of exposed yourself to over the years. I guess how would you advise them to figure out what's right for them? What do you wish you knew when you were that young kid who started at Dartmouth?
A lot of things. I wish I could have learned from my mistakes back then. But I'd say that the one thing is, people often say, follow your dreams, follow your heart, things like that. And for me, that's been part of it. But I think probably in my mind, it's been more follow the things that I feel that I can persevere with for the longest. Because often it's, inspiration will come and go for a particular endeavor. And yet if it's something that you just believe in, you have some conviction around, that's something that will get you through the more difficult parts. I think that, following dreams and what you like hope for, that's something that can certainly change with time. But if it's something that you feel, in my case, the sort of, I spent 10 years doing the MD PhD. That was something it was it's a lot of persevering there.
Yeah, you did it.
Deciding to leave Deepak's lab and start Wildtype. That's something that, just follow the things that you feel good about committing to, and for the long term. And if they work out, then great. And if they don't, I think that's when you don't have regrets because you gave it everything.
That's so well put. Arye, it's a pleasure to talk to you. I'll let you go to your meeting, and we'll pick this up some other time.
Thank you so much for having me.
You've been listening to Season 3 of Plenary Session. Plenary Session is produced by Kiana Klossner. Music by Ian Straley and Audrey Tran. The views expressed on Plenary Session are those of whoever said it and no one else. Plenary Session is not medical advice. Follow us on Twitter at plenary_session. Until next time.